Hello, everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by WuXi AppTech. Today's topic is Drug Discovery Innovation, Target Deconvolution and Tractability Assessment. We're so delighted you joined us. I'm Uduak Thomas, Senior Editor at Jen, and I'm delighted to be your host for today's webinar. Let's meet today's speakers. Dr. Zhang Yao Ma is Senior Director, Head of Target Discovery and Validation and Peptide Discovery Platform for WuXi Biology at WuXi AppTech. Dr. Wen Zhang Chen is Senior Director, Head of Biomass Spectrometry Platform for WuXi Biology at WuXi AppTech. We'll get to their presentations momentarily, but first, a bit of background on today's topic. Target deconvolution, particularly when paired with proteomics techniques, plays a crucial role in drug screening, covalent drug discovery, mechanism of action discovery, and target identification. By using chemical proteomics, researchers can more precisely identify interactions between small molecules and cellular targets, aiding in the development of new therapeutic candidates. So in this webinar, our speakers will explore cutting-edge drug discovery techniques for target deconvolution and multiplex target discovery. They will also delve into how multiplex target discovery and target tractability assessments accelerate the drug discovery process. Now, during the webinar, you'll learn how to apply target discovery and target deconvolution technologies in the early stages of drug development, enabling faster, more accurate identification of viable drug targets. Additionally, they will discuss the use and value of DNA-encoded libraries for target tractability assessment, and you'll also learn about the technical strengths and service opportunities that are available through WuXi AppTech. So what are some key takeaways from the webinar? Well, you'll learn about accelerating drug discovery and mechanism of action studies through proteomics and target deconvolution. You'll learn how to apply multiplex target discovery and deconvolution techniques to identify promising drug targets. And you'll learn about leveraging DNA-encoded libraries for efficient target tractability assessments. Now, at any point during today's webinar, if you have a question, please feel free to enter it in the Ask a Question panel on your screen. All right, let's get started.
Hello everyone, I'm Wen Zhang Chen from the Biological Department of WuXi AppTech. I'm pleased to welcome you to this presentation. Over the next 20 minutes, we will be exploring MS-based drug screening technique and proteomics technique. Please also feel free to ask questions for the QA and at the end. The mass spectrometer and technique and the proteomics technique have been widely used in biological research. The techniques are also useful for earlier stage drug discovery, such as drug screening, mechanism of action discovery, drug target deconvolution, and rule out problematic compounds, also biomarker identification. Today, discuss MS-based drug screening technique and proteomics techniques, also their application in drug discovery. I will also present some internal case examples on drug screening and drug target deconvolution using chemical proteomics technique. The first technique I would like to discuss is the affinity selection mass spectrometer technique, ASMS technique. ASMS is a high-throughput drug screening technique. First, the compounds are incubated with the target protein. Then a separation step is performed. Then the non-binders are then washed away while the binders are retained and identified subsequently by high-resolution mass spectrometers. Compounds in the semen pool can be identified by their exact mass and chromatography retention time. Mass spectrometers' high resolving power allow hundreds of compounds to be mixed as one pool and screened together, greatly enhancing throughput. The ASMS technique is a binding affinity assay. which is similar to DNA encoder library screening method. SMS use a high-resolution mass spectrometer to encode and decode compounds. DNA encoder library use DNA sequence. SMS is a label-free method, which is compatible with any drug-like compound library. In contrast, DNA encoder library requires all the compounds to be attached to DNA sequence. This is the advantage of SMS method. Covalent drugs have gained popularity in recent years. Covalent drugs are a class of active molecules that bind to a drug target irreversibly by forming a covalent bond between the active molecule and the target. Compared to reversible drug molecule, covalent molecule have the advantage of low dose and high potency and can target previously undruggable targets. However, drug developers in the past avoid covalent drugs mainly because of off-target binding and the subsequent risk of toxicity issue. Thanks to advance in chemical proteomics, off-target binding can be evaluated and avoided in the earlier stage of drug discovery. This is an overview of the history of covalent drugs. However, these drugs were not discovered by design. The covalent mechanism was elucidated after it was found that the drug were effective. In addition, phenotypic screening is a popular approach for first-in-class drug discovery. One disadvantage of phenotypic screening, however, is the challenge of finding drug targets. Covalent drugs allow drug targets to be found using proteomics method with much less effort. This is also another advantage of covalent drugs. KRAS is a popular anti-tumor target that was previously considered undruggable. KRAS G12C is one of the top mutations in many types of cancer patients. As the mutations will result in tumor growth. In this study, we selected KRAS G12C as our target to evaluate the performance of our covalent drug discovery platform. These are two marketed KRAS covalent inhibitors. Our intact mass and peptide mapping results demonstrate that a covalent bond was formed between the drug and the KRAS G12C. The binding percentage was nearly 100%. However, it was unclear whether this bond can be formed within cells or in vivo. To address this issue, we employed target proteomics to gather information. As we can see, a clear dose response curve was generated, and the GC50 of the two compounds is around 10 nanomolar. This result suggests that both compounds are highly potent. Target proteomics is an essential assay for evaluating the efficacy of the covalent drugs. The method can also be used for in-cell covalent screening. The advantage of in-cell covalent screening is that we don't have to worry about protein activity. Sometimes it can be difficult to obtain high-quality active and purified proteins. This is our in-cell covalent screening workflow. In the past few years, many projects using this workflow have been successfully completed. One concern with covalent drug is off-target binding. We worry that off-target binding will result in toxicity issue. This is also why covalent drug was avoided in the past. To address this issue, chemical proteomics technique was employed to identify all off-target covalent binders. This is a competition assay in which free cysteine can be enriched and quantified by mass spectrometer. If a cysteine reacts with covalent drug, it is blocked, cannot be enriched or analyzed by mass spectrometer. By comparing DMSO group and drug-treated group, we can determine which cysteine peptides are free, which are blocked. In other words, we can determine which cysteine residue form covalent bonds with inhibitors. The right panel is an example. The same marketed covalent inhibitor was used as the tool compound. The result shows that there is only one statistically significant result. This means that there were low off-target binding in our data. Additionally, a clear dose response curve was obtained. The GC50 is close to the result of the previous assay, which is also 10 nanomolar. Protomy-wide covalent fragment discovery can also be achieved by combining this method with a covalent fragment screening approach. The method can obtain binding affinity information for all proteins with each fragment. Unlike single protein analysis, proteomics is a technique that can identify and quantify all the proteins at a cellular level. We can quantify up to 10,000 proteins in less than 1 hour. This makes the technique very attractive. We use Simon Karas covalent inhibitor from the previous slides as the tool compound to evaluate our platform's performance. The result shows that many proteins associated with Karas pathway were regulated, and a clear dose response curve was achieved. The GC50 was also around. The GC50 is also close to the result of the previous assay. It's also around 10 nanomolar. Suggesting that proteomics profiling result can be used to evaluate drug efficacy and as a universal screening tool. In addition, global proteomics is also compatible with reversible compounds. Also, as we can see, FOS1 protein and some other protein regulated by FOS1 were downregulated upon treatment. A recent science report also reached a similar conclusion, but at the transcriptomics level. This also suggests omics techniques are reliable across different labs. Post-translational modifications play an important role in understanding cell biology and discovering methods to treat and prevent disease. Additionally, only a small portion of drug affect target protein abundance. However, many drugs directly change PTM abundance. For example, kinase drug change phosphorylation abundance. For Karas inhibitor, we expect ERK phosphorylation will be changed after treatment. Previously, Western blot was used to quantify ERK phosphorylation and to evaluate Karas inhibitor efficacy. In this slide, phosphoproteomics instead was further employed to elucidate the drug action mechanism and evaluate drug efficacy of Simon covalent inhibitor. The result shows that Karas inhibitor treatment significantly downregulate ERK phosphorylation. This is what we expect, also indicate the drug were effective. Also that many other proteins exhibit a significant change in phosphorylations. The affected pathway are shown in the right panel. The results are also similar to those in some recent publications. Compared to Western blot, phosphoproteomics can provide a system-wide picture. At WuXi, we support proteomics for phosphorylation, glycosylation, ubiquitination, methylation, and acetylation. This is drug-resistant case example. Antibody drug conjugate discovery is at the forefront of cancer treatment. However, many cancer patients will inevitably develop resistance. In this case, proteomics was employed to help elucidate resistant mechanism. The proteomic profiling comparison between normal cell line and resistant cell line shows that target antigen expression level decreased by 30 times. Also, a larger protein has 1,000-fold increase, which may affect the internalization of ADC. The KGG enrichment also showed that lysosomal dysfunction may cause the resistance. This slide is about drug-targeted deconvolution using proteomics. A drug compound can typically bind to multiple proteins. Therefore, it would be beneficial to identify those proteins in the earlier stage of drug discovery. The information would obviously reduce the later stage risk and cost. Additionally, after phenotypic screening, we have active candidate compounds, but we don't know which protein is the interaction protein target. So we need a targeted deconvolution technique. Chemical proteomics can be used for targeted deconvolution. A affinity tag and a photolabeling group are attached to the active candidate compound as shown in this slide. The photolabeling group will covalently link proteins in close proximity, then the modified compound is incubated with cells. The proteins that binding to modified compound are cross-linked and be enriched. The mass spectrometer is subsequently used to quantify interaction proteins. Quantitative proteomics is further used to reduce non-specific binders. A pan-kinase inhibitor was used as the tool compound to evaluate our platform's performance, and over 30 kinases were identified, which prove the feasibility of this technique. Thanks. That's all of my today's topic.
Hello everyone, this is Zhongyao Ma from WuXi Biology In Vitro Biology Unit Early Discovery Platform. Today it's my honor to introduce our capabilities regarding integrated target discovery platform. Today I will mainly focusing on our target discovery, target deconvolution, and the target validation capabilities. So before we actually run into the details of our platform, let's take a look of some of the highlights of our integrated target discovery platform at WuXi AppTec. So within WuXi AppTec target discovery platform, we already have more than 1,800 different animal model data sets. And we have been already successfully validated and discovered more than 100 different targets for our clients in the past 10 to 15 years. And of course, the speed is always the key thing. We have more than 50 plus in vitro and in vivo screening models that is ready to use so that our clients can start their experiments and projects anytime. So why target discovery and target validation so important in early drug discoveries? So we all know the drug discovery process takes a very long time, ranges from 5 to 15 years, starting from target identification all the way to the lead optimization until we can get a very promising candidates to follow up. So we spend a lot of time, a lot of money into this process, and we want to make sure the target is a very good target that was followed up. Otherwise, we are spending a lot of time and money into nowhere. But what actually makes a good drug target? So we think a good drug target should contain at least following 6 different properties, including such as druggabilities, which means you can find some potential compound that can bind into it and modulate the functions. And also, it should follow a distinct body expression patterns in a certain disease so that it's not uniquely expressed all everywhere, which means it's potentially to be toxic and dangerous. Of course, you also want to make sure your target is IP favorable so that you can have some potential space to make and looking for novel chemical identities. The good news is starting from 2010, from the past 15 years, we are seeing that there are more and more drug targets showing up in the market and also in the academic field, which means there are more and more targets could be potentially used to cure a certain disease, and the process is still going up. So why choose WuXi AppTec for target discovery? So of course, the first thing is that we have so many rich experience regarding target discovery. In-house, we have more than 1,300 different cell lines that is ready to use, so that it saves you a lot of time and money to actually not really need to construct a new cell lines for your experiments. These cell lines covers more than 28 different cancer subtypes, and also, of course, we are covering other disease types such as immune disease and the metabolic disease. And also, we have more than 50 plus ready-to-use Cas9 stable cell lines, so that we can directly using these cell lines to do gene manipulation experiments. And this number is keep growing up. And we are not only focusing on in vitro screenings, we are also focusing on in vivo screenings. So we have more than 45 different in vitro and in vivo models that is ready to use, including a lot of really good models in mice. Now, of course, the data and the quality is always our first priorities. So all the cell line characterizations, all the cell line validations, all the in vivo models validations are all SOP guided so that we can make sure, although the target discovery experiments and projects is always the tricky stuff, but we can make sure all the data was with high qualities and high speed. So the first part, I would like to introduce our capabilities of target discovery. In these sections, we will briefly introduce how we do in vitro screenings and how we do in vivo screenings and show some of our case studies to walk you guys through how we do target discovery. So the first case study is I want to show you guys how we're using in vivo screenings to looking for potential new drug targets related to tumor growth. So in these studies, we actually first generate a gradient host immunity pressure models. So starting from newt mice, it was basically immune deficient mice. So there's no immunities at all, no T cells, nothing. And then we goes into the wild type mice, which means it's basically normal immunity pressures. And then we also add in the immune checkpoint PD-L1 and PD-1 drug to make sure that we have increased the immune pressures in these certain models based on the wild type mice. So we transplant these MC38 cancer cells into these in vivo models. And basically on the right part, on the graph B, we can see based on different immune pressures, these tumors are growing at different speed, which means our immune pressure models was basically working. So after the mice, the tumor grows for certain days, we take out the tumor, get the tumor cells, and goes for those potential gene candidates. What's interesting to us is that we do find some genes that was enriched in a certain in vivo models. For example, let's take a look on the right paragraph. We do find some PD-1 independent gene candidates that shows up not only in the wild type mice, but also even we put into PD-L1 and PD-1 drug immunotherapies, these genes are still enriched, which means these genes are helping the tumors to get resistant to the immune system, even with the PD-1 and PD-L1 present. So we found in total about 23 PD-1 dependent genes and about 30 PD-1 independent genes. So these genes are going to be our follow-up potential candidates, but they are still too much to do the target validations one by one. What we're going to do? So now we have our potential candidate genes, which is the ANKRD52, and we want to perform the in vivo competition assays and also in vivo validation assays to make sure we actually do identify some promising hit genes from our models. So in order to do this, we actually designed 4 different individual NKRD52 sgRNA together with 3 non-target sgRNA controls to perform these assays. So the first thing we have been seeing is that after we do use the ASGN-KRD52, we see a significant growth disadvantages of the tumors in a wild-type mice, or let's say a immune-competent mice, which is clearly can be found in the left figures. And also, this is also validated in the in vivo mice models, as we can see on the right figures. No matter we do the ANKRD knockout 1 and the ANKRD52 knockout 2, the tumors always grow slower comparing to non-target sgRNA controls. So that means these cells, after you knock out the ANKRD52, these tumor cells grow slowly if there's no immune pressures. However, if we move these models and these cells into a wild type mice, or let's say immunocompetent mice, an interesting thing that we find is that now these ANKRD knockout tumors grows at the same speed as the non-target sgRNA control tumors, which means this gene knockout give them growth advantages in the immune-competent mice. A second case I want to show you guys is talking about the synthetic lethal screenings in drug discoveries. So the synthetic lethal is not a very novel idea. The concept of the synthetic lethal was first proposed in 1946. And after that, it was basically takes around 40 years to make sure this synthetic lethal was actually can be proved to be durable. And then after 2011, there's so many different synthetic lethal pairs was under investigations, and in 2017 and 2018, several synthetic lethal pairs was finally get approved by the FDA, and this concept has been widely used in the disease therapeutics. So on the right figure, we can see there's a lot of hot synthetic lethal pairs, which focusing on, for example, like ARID1A, TP53, BAP1, and all other genes. So there's still a lot of new synthetic lethal pairs that was waiting there to be discovered and could be potentially used to treat the cancer disease. So how we do the screenings and how the synthetic lethal works. So basically, we first construct a knockout cell lines, which contains the first oncogene knockout, and then we do the whole genome library transfections or transductions, and then transplant these cells into the mice models with different immunity pressures. And after several days, we monitor the tumor growth day by day until we determine, we see a significant difference of the tumor volumes within different experimental groups. We collect these tumors and looking for whether there's a specific gene was particularly enriched. And that could be our potential synthetic lethal pairs. On the right part, it shows you one of our examples of our synthetic lethal screenings. In these screenings, we perform in vitro screenings and using an epigenetic focused sgRNA libraries. And after screenings, we identify 12 different genes was enriched by using these in vitro screenings, and one of them was SMARCA2, which has been reported as a synthetic lethal pair to our oncogene SMARCA4. So these technologies and these case studies showing that our workflow works, and it could be applied to potentially other genes and other cancer models. So basically, in this part, we show how the in vivo screenings can help to identify potential key modulators of cancer. For example, in our case, the ANKRD52. And in the second case, we're showing how we can use in vitro and in vivo models to screening for potential synthetic lethal pairs and looking for new genes. And also in WuXi AppTec, we can provide whole genome based screenings to increase the potential possibilities of identifying a new target, which also help you to go into not using a focus library, which is potentially biased. So for the next part, I want to introduce how we're going to use our platform capabilities to do the target deconvolution. So why we need targeted convolutions, why this is so important and popular within these days. So basically, there's two main strategies to develop potential promising hits. The first one was basically we all know the target-based drug discovery is you first identify a target, you purify the proteins, you construct the cell lines, and then you do the screenings using a real, compound libraries, no matter it's small moleculars or, peptide or antibodies. And then you got your first hit, and then you do those optimizations. But sometimes you don't really have the target. You're starting with the disease-related models, either cell models or animal models, in most cases, cell models. You do a phenotypic screenings. Now you get a very promising hits, which can rescue the phenotype of the disease cell types. But you don't actually know what this compound actually binding to, or let's say what's the actual target. So then you're going to need the target identifications, or let's say target deconvolutions, to help you identify the actual target so that you can go for the downstream MOA analysis, pathway analysis, and also lead optimization strategies. So what's popular in the commonly used technologies in target deconvolutions? I think there's mainly three different strategies that could be used for the target deconvolution, including expression manipulations, chemical proteomics, and also the proximity-based labeling strategies. So here we want to show you guys that, how this target deconvolution could be used to facilitate the targeted protein degradation drug discoveries. So in this case, we first start with our WuXi internal molecular glue focus libraries, which contains around like 5,000 different unique design potential molecular glue compounds. We construct two different cell lines, just like you guys can see on the right. The first one was GSPT1 hybrid cell lines, and the second one was GSPT1 hybrid with a cereblon knockout cell lines. So we want to looking for both cereblon dependent cells and also cereblon independent molecular glues. After the first screenings, we identify around 142 primate hits, and 47 of them can be confirmed in the secondary screenings. And the 14 of the hits shows very nice dose response, and 4 of these 14 hits was actually non-cereblon dependent molecular glues, which means they show a very significant GSPT1 degradation effects even in the cereblon knockout cell lines. As you guys can see on the right, we show two representative data pictures. One of the glue, which is cereblon dependent, the other one was basically cereblon independent glues. So how are we gonna, using target deconvolutions to deconvolute the potential E3s that are involved in these pathways if it's cereblon independent molecular glues? Basically, the first strategies you can use, we call them proximity labeling approaches. Basically, you construct a cell line which have a fusion proteins, contains your protein of interest, but also a biotin ligase connected to it. So this biotin ligase will biotinate any binding partners get close enough to the protein of interest. As we all know, to make a target protein degradation happens, the glue or the PROTAC has to recruit an E3 ligase, get close enough to the protein of interest. So using this proximity labeling approaches, the biotin ligase can biotinate the NAE3 ligase that get close enough to the POI, and then we can using proteomics to actually identify the E3 ligase involved in this particular molecular glue. So on the right case studies, we use a reference compound called ARV110 to see how this technology works. So basically, first of all, we identify a very significant, cereblon enrichment, which is what we expect, and also we identify several, genes or let's say proteins was highly enriched, which can be identified as the POI's natural binding partners, which means they're gonna get close to the POI no matter if there's a glue or PROTAC or not. And also we have been identified several other E3 ligase, we call them E3 ligase X and E3 ligase Y. They can be identified in the proteomics, which could be also involved in the ARV110 treatment and and of course the cereblon is the top one that has been identified in this POC studies because we are using a reference compound. But actually for all other potential unknown compounds, using these strategies, you can identify a bunch of potential E3 ligase candidate list, and you have to do further experiments to actually validate whether these candidate genes do works in the target protein degradation pathways. So what we're gonna do, basically, if you see these, candidate genes, what you're gonna do is actually you could do like for example anti-array library transductions to manipulate these genes one by one in a plate format, so that you can see which genes is actually controlling the degradation effect, and that's the one that the actual target that involved in target protein degradations, or let's say the non-cerebral dependent degradations. So, but target deconvolution could not be only used in the case studies we have been talking about today. Can be also used for other perspective. For example, you can use it to identify novel molecular glue or degrader targets, not only for the E3s, but also looking for a new novel degradable target. And also, we only covers the mainly cancer-related therapeutics in the target discovery part, but actually the target discovery strategies could be applied into other disease scenarios. For example, the metabolism-related target discovery is also a very hot topic and could be used. So in summary, if you have an assay that could be, used, for example, like for sortings, for selections, it could be transformed into a potential target discovery assays to looking for new drug targets. And the last part I want to talk about is something a little bit new, but we are still working on, which is the target validation using cell painting-based MOA profilings. So the cell painting was a pretty cool technology. It was basically high content image-based approaches to directly recording the morphological changes by using a multiplex fluorescent dyes. All these fluorescent dyes are targeting different cellular features. So basically, after standings, you're going to get so many informations directly by looking at the images of these cells. And then you can comparing the before drug, after drug, so that you can see whether these drugs actually make some changes to the morphologies of these cells. And these cell paintings can not only be used for the drug screenings and can be also used to toxicity, safety evaluations, assay activity predictions, identify potential drug targets. And today we're going to talk about how we're using cell paintings to predict drug MOA. So first of all, if you want to using this technology, since this is a phenotypic-based screening methodologies, you want to make sure this data was highly reproducible, or you can see the same effect from batch to batch, and you can see the same effect from the same compound treatment with the same dose. So on this slides, basically we can take a look on the blue box and the green box. Basically, we're using two different compound treatment. We're using four different plates for the same compound. We do these experiments in different batches, different days, and you can still see after the cell painting experiments, we using UMAP to analyze the picture features and do some clusterings. And these compounds can still be clustered together, even in different plates, even in different batches. So this tells us that even this is a phenotypic-based selection strategies, it's still highly reproducible. And then we want to using this, stable technologies to take a look on actual compounds to see whether we can use these technologies to do MOA profilings. So to begin with, we pick several different, pathway inhibitors, for example, like high-tech inhibitors, kinase inhibitors, PDE-related drugs, and the apoptosis-inducing drugs, those kind of stuff. And interestingly, we can see for the high-tech, for example, for the high-tech inhibitors, 2 out of 4 can be actually clustered together. And we see the same thing for other, drug class, for example, like PDE drugs was also clustered together. And those drugs that are known to have a mitochondrial effect also can be clustered together. So basically, in our experiments, 17 out of 22 compounds, actually the clustering patterns aligns with our annotations, which means we know their pathways before we do the experiment. We predict they should be clustered together, and after the whole experiment, by directly using the phenotypic images, we can see these compounds get clustered together. So this result tells us that the cell painting technologies could be potentially used for as the MOA prediction studies if we already have so many reference database in-house of each pathways, and we know how the morphology gonna looks like after we change or inhibit or activate this particular pathway. And to end up, I want to go through the big pictures of our platform. So basically, we do target identifications, target validations, and also target trackability assessment. So for the target trackability assessment, we're mainly focusing on analyzing the trackabilities of the potential compound in a high throughput way. For the target identifications, we do either target discoveries if there's no ligand available. So we can also do target deconvolutions if we already have a ligand coming from a very successful phenotypic screenings. And of course, we have a huge flexible target validation assay packages to support the downstream target validation experiments. So in our platform, we have been, found a lot of interesting targets with our clients and with our collaborators in the previous 10 years. And here, this slide showing some of our selected successful stories. And thank you all guys for your attentions. And I'm really happy to answer any potential questions. Thank you.
Many thanks to our speakers for their informative and interesting presentations. We're going to begin the Q&A session in just a few moments, so please submit your questions now if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours, so please share the link with your colleagues who might not have been able to tune in today.
I have also noticed that some great questions coming from our live audience. Let me take a look at the question. The first question is whether affinity selection by spectrometer can be employed for peptide screening. The answer is yes. Recently, there were some publications on peptide screening by combining ASMS technique with nano LC-MS technique. Ultra-large chemical libraries was screened to find the high-affinity peptide binders. The technique is capable of identifying binders from a library of 100 million randomly synthesized peptides. The advantage of ASMS is that ASMS is a label-free method, support many different types of amino acids. The second question is that whether post-translational modification proteomics can be employed for drug-targeted deconvolution. The answer is also yes. For drug-targeted deconvolution, multiple techniques are needed. PTM proteomics can help elucidate drug action mechanism and can consequently help identify drug targets. The third question is that what is the disadvantage of chemical proteomics for drug-targeted deconvolution? Also, how to minimize the non-specific binders. One main disadvantage of chemical proteomics is that affinity handle has to be attached to active compound. This modification will sometimes result in significant activity loss. So we recommend to assess the modified compound activity before proteomics part. Also, to minimize non-specific binders, we recommend to add a active control. The active control also have an affinity handle and a photolabeling group. And the comparison between active compound and the selective compound can avoid non-specific binders.
So I do see a question asking about how to choose between the in vitro target discovery screening and the in vivo target discovery screening. I think this is a pretty good question. So in oncology area, it was pretty straightforward. If you have a co-focus library, the in vivo screening models will give you more corresponding informations which more closely related to how the tumor actually grows under different immunity pressure situations. And the in vitro screenings can give you a higher potential candidate gene pools, so you can search for more potential genes. But for other disease area, it might be a little bit different. For example, in the metabolic disease area, we find out that a lot of readout is pretty consistent and stable for the in vitro cell line assays. However, for the in vivo studies, it's going to be a little bit more complicated to perform natural screenings. So in those kind of cases, we would recommend our client to do in vitro screenings because we have ready to go assays. The assay was pretty consistent and the readout is sortable or screenable. Thank you. A second question is asking about how the target deconvolution can be used in other therapeutic area besides target protein degradation. I think this is also a pretty good idea. So for the target protein degradation area, the target deconvolution is very important. You can use it to deconvolute those potential E3 ligase that is involved for the protein degradations, and so that you can do further SAR analysis and the MACM analysis. However, the target deconvolution can also be applied into other area. For example, if you have a direct binding compound, you can still use in target deconvolutions to deconvolute the direct binders of this compound so that you can further purify the proteins and then do downstream SAR and MACM analysis. Also, you can also use target deconvolutions for other potential purpose. For example, you can use target deconvolution to direct take a look to see how the compound not only binding to its potential target, but also do it bind to other uh, off target, like the target in the same pathways, the target in the same super families, and also it can give you a lot more informations besides its actual real target.
And that's all the time we have for today's webinar. Before we go, one last reminder that this webinar is going to be available on demand in the next 24 to 48 hours. So you should get a link in the email you used to register, and we invite you to share the link with your colleagues who might not have been able to tune in today. I'd like to thank both of our speakers for their presentations. I'd also like to thank today's sponsor, Wuxi AppTech. And I'd like to shout out the superb multimedia group at Sage for all of their support. Most of all, thanks to every single one of you for watching today. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.